Norbert Kruger from the Maersk McKinney Moller Institute in Denmark, and he's come to speak to us about from early vision to symbols.
Thanks a lot. So, the main part of my talk will be that I will talk about this paper, which has been published last year in Kruger, and actually one of the authors is Sinan, who has just given the talk. So there's a story behind this paper, because what we wanted to do in this paper is we want to summarize 2 days' knowledge about the human visual cortex for computer vision scientists. So we wanted to basically cover the literature and see what has happened the last 20 years and to formulate in a language that is digestible for guys like you. Because probably if you read one of the specific papers that they publish, it's very difficult to dig through there. And for that, we have quite a different kind of scientist. Peter Janssen, he is a neurophysiologist in Leuven. Sinan Kalkan has just given the talk. Markus Lapp is a psychologist in Münster. Alice Leonardis, probably many of you know, he's a machine learning guy. Justus Piazza, he's a modeling guy, Antonio, and Laurens, he's a theoretical neuroscientist. So it's a good mixture of people. But I want to start with some annoying prior remarks. And so the whole story was actually to say, okay, it is the human visual system has a deep hierarchical structure, and it is worth going that way. But coming to this annoying prior remarks, so there has been the work by Google and Wiesel, who got the first idea what might go on in areas like V1 in the visual cortex, and they got the Nobel Prize for that in 1981. And pretty much motivated from their work, there was the famous work by David Maersk, and that actually made quite a link between biological motivated vision and computer vision. So there was quite a fruitful cooperation. And what Maersk did was basically also introduce some kind of a hierarchy. You have some kind of edge processing, then you have this kind of 2.5D viewing, and then you have kind of an object-centered representation. You can think about a 3-step hierarchy, so to say. But one needs to say that this approach failed in some way. So computer vision went more in the area of machine learning, and I think when we want to refresh this link between biological vision and computer vision, we need to reflect on why has it failed. And I think there are 2 reasons why it has failed. So the one is at Maersk time, so there was just a lack of knowledge on low-level visual modalities like optic flow, edge detection, stereo. All this has developed in the last 20 years, and we are now on a completely different level there. The other thing is at Maersk time, computers looked like this. And this was just not good enough for trying to model the human brain. So, and also there we are in a completely different situation right now. So I think this can give us hope to think about that. So as I said, in the 80s and 90s there was a quite strong link between these different disciplines, but this link has been diluted from both sides in some way. So in some ways, so computer vision was not interested. Probably they said, okay, it's just not good. We can do something better when we do just machine learning. And also from the neurophysiology side, they are, in this I think it's still a problem. They've given up to trying to understand what is going on in the brain on a functional level. So they're very much in the details, what neurons are there and stuff like this. But there was kind of a lack of trying, okay, what are the algorithms? What are the principles behind it? So that leads that this term biologically inspired got a somehow bad reputation. First, it is not efficient, and also everything started to be biologically motivated. So if you used Gabo wavelets, then you had already a biologically motivated system. Okay, and the authors here of this paper, they thought, okay, we have a better situation, a restart might be worthwhile, so there's a much better understanding of early vision. there are more computational resources, and there are also still many unsolved problems in computer vision. And the aim of this paper is to distill the essential knowledge on the human visual system for engineers. Okay, and this was the motivation. Now I basically present the content of the paper now in the next 15 minutes. Starting with some basic facts. So when you look at the cortex and you flatten it on a table, it's kind of a half a meter piece of cloth or something like this. And all the areas that are colored here have to do with vision. Meaning that 55% of the neocortex of the primate brain is concerned with vision. And what is relatively well established is that there's a number of areas in which the visual information is processed, from the retina to the LGN to V1, V2, V3, V4, and then it goes into ventral pathways and in the dorsal pathways with sub-areas. Actually, the size of these areas here is proportional to their size in the brain. The idea of brain maps is actually a quite old one. So this is from a Dr. Alisha Sivata in the late 18th century. So that was his idea. So he had already the ideas of brain, and there were areas for integrity, for dignity, and fear, and stuff like this. So it was full of areas being responsible for different stuff. And also they said, okay, vision has some importance, and the visual areas here. So it's the small areas between the eyes, and why it is small, because it's such a simple problem. So I think that this is well established now. We have gained some knowledge there. So, and what I basically now want to do is I want to go with you through half of the brain in 15 minutes. So this is also a table from this paper. So I will not explain the whole table, but the idea is also that computer scientists, machine learning guys, they can look up basic facts, and they don't need to look in different papers. They can look in one paper, and then of course they are reference to specialized work. So here are basic facts about the different visual areas I talked about. So retina, LGN, V1, V2, and so on. All the different areas that I've shown before. That information is given about their size in square millimeter, the receptive field size, at a certain degree of peripheralness in the visual field. Then there is something about latency. So how long does it take from a signal coming to the retina until the neurons are reactive? And here you see, so in the retina it takes 20 to 40 milliseconds. Then in V1, 30 to 40 milliseconds, V2, 40 milliseconds, 50, 70. So it's approximately 10 milliseconds per level in the processing. So this is a strong argument about the hierarchical structure. You basically see how so that with every level it takes another 10 milliseconds for the information flow to go through. Okay, and now what I want to go is I want to go basically from the retina to the ventral and dorsal pathway and briefly indicate what people believe what is processed there. So we are now talking about the retina and the LGN. So this is all figures are from the paper. So what we did was that we showed the receptive field size of the neurons depending on where in the visual field they are positioned. Normally, the receptive field size of neurons in the periphery are much larger than in the fovea, but that's not the case for the first level of the retina. So there, it's the same receptive field size for the rods and cones. And what we have is on the input side, we have basically we have two types of sensors. One is roughly comparable to RGB sensors on a chip, and the other system that is responsible for different shades of gray, to code different shades of gray. So that's the input sensors of the retina, and the output of the retina is that a transformation where color is coded in terms of opponency, so a yellow-blue opponency and a green-red opponency, and where contrast is coded by means of these center surround cells. So that's the first step, going from pixel-wise information to this kind of color opponency and contrast. Then it goes to the LDN, so there's not much feature transformation going on there, but what is done is that the neurons are, that the input is ordered so that you have neurons from the left visual field and right visual field close together so that you can find correspondences later in V1 to get stereo information. And the receptive field sizes are also slightly larger. So now we're going to the cortex here. And here we have 5 areas, V1 and V2, which are really huge areas compared to the rest. I mean, the whole V1 is larger than the complete dorsal pathway. And then we have V2, V3, not much is known, therefore we did not describe it in the paper. It is known that it is there, but no one knows really what it is for, V4 and MT. So something about the occipital cortex, these are these yellow areas I'm talking about. So it is a very large area, so it's 70% of the visual cortex actually. And what is in general believed that there is task-unspecific generic scene representations. So these are not task-specific representations that are built up there, but generic representations. So 70% of the cortex is concerned with these issues. What is also typical for all areas in the occipital cortex, there's some kind of retinotopal mapping. So here you see the retina, and here you see how it is mapped in the cortex. So the retinotopal structure is preserved. Then what you see again, here in V1, for every area we show the receptive field sizes, so they are larger than in the LGN. And so what we have is, we have this double opponency. So we have contrast cells, or neurons responsive to contrast, but at the same side to color opponency. We have kind of edge detection, different kind of edge detectors. We have point feature detectors in some way. We have disparity, and we have optic flow where the aperture problem has not been resolved yet. So here the computed flow is still orthogonal to the edge orientation, so to say. V2 is some kind of a copy of V1 with larger receptive fields, but while here features these features can be computed locally, we have now additional components, for example, like border ownership. So to code to which object does to which side does this edge belong? We have relative depths in contrast to absolute depths here. We have also something illusionary contrast like in the Kanitsa triangle, and we have also edges that are coded by which are not coded by contrast. So these are to a large degree features that require some kind of more global scene interpretation. Otherwise you don't come to a stable assessment of border ownership, or you don't get this illusionary contrast. And it's interesting that V1 and V2 actually have very similar size. It seems to me it's some kind of copy where some additional features are processed that require some kind of global reasoning. In V4 we have then decide curvature in color coded, but here color is coded really as an object property. So at this level we become independent from the illumination conditions and can assign color as an object property. MT is a small area here where 2D motion and 3D motion features of various kind are computed. Transparent motion, here we see the aperture problem has been resolved actually here, but also different kind of 3D motions. Okay. So this occipital cortex is shared by the ventral and dorsal pathway. So I'm now talking about the ventral pathway. So there actually, this kind of division in areas is debated. There are many different ways of doing that, and some people also say it's a more kind of a continuous change of representation. So it's only 17% of the visual cortex. It is assumed that it is important for object recognition and categorization. And as I said, there are many suggestions to how to divide it. So, but we said, okay, this is kind of what is done often. You have one area TEO and one area TE. So that's a common distinction here. TEO cells actually they have a quite small receptive field. It is already much more centered in the fovea. As also for TE, we have kind of combined features, shape and color features. And in TE, people believe that there are some kind of semi-complex features that are still below the object level. An exception is faces, so faces seem to be coded there as well, but in general, these features seem to be below the object level, and I want to give you an indication how they come to these features. So this is a work by a guy called Tanaka, and I must say also that his work is not undebated, and I guess the experiments or how he has derived these features gives an indication why it is debated. So he basically took a visual stimuli and then he looked for a neuron in IT that was responsive to it. Yeah, so you can measure responsiveness of neurons that they fire, that they're very active, that they fire with a high frame rate, so to say. What he's then doing is he takes the stimulus and simplifies it and sees whether the neuron is still active. And then he goes a step further in simplification and looks, is the neuron still active? And then he tries to simplify it more, and then the neuron's response is not active anymore. And then he says, okay, this neuron codes this kind of feature. And of course, there's a lot of, let's say, experimental decisions in this kind of simplification process. But just to give you an idea how these icons here are derived, and of course there are many more than these four. Talking about the dorsal pathway, also a rather small area, maybe 50% of the visual cortex. It is much less known about it than the ventral pathway. Many more distinguished small areas, and here action relevant information or related to space and position in space, this is coded here. We have MST and CIP as two areas that still do some kind of generic processing. So here we have Q-invariant 3D shape information in CIP, and MST basically codes the ego motion of the observer. 10 minutes, yes, that's fine. And then we have four areas in AIP, handshake and affordances seem to play a role. MIP is about reaching. We interestingly see that the receptive field sizes are very different. So here you have much larger receptive fields, which makes a lot of sense because reaching is a more global kind of activity. In VIP, the ego space around yourself, the human is coded, and LIP is connected to saccadic eye movements. This is also from the paper, so Lawrence-Biscott actually, they took all these icons that we have derived to visualize this kind of processing, the receptive field sizes here, the area sizes, and the connectivity that is there between, and then basically combine it in one big figure. So here you see the visual cortex in one figure in some way. And the idea was also to make it educational, of course. So to not go too much in details, of course, this also means simplifications, but at least then to give intuitions. Okay, where does language come in? At the higher levels of the processing, for example, when we talk about color as object properties, this can be associated to 4 objects are coded as probably combinations of these kind of descriptors, so one is not enough for that. Prepositions, spatial prepositions here could be associated to these kind of areas, and action and verbs could be associated to these areas. And of course, the simple grounding problem is, okay, how is all this linked to the stuff that is going on below? And a lot of visual information is relevant for linguistic is in the temporal structure. And the next video, I just want to show that I want to make sure that we are mainly doing technical stuff, so that you don't believe that we only think about the brain. So this here you see our visual system in a cooperation with the University of Göttingen, Florentin Burkharter's group, and you see that we do this object detection and post-estimation in rather complex scenes. And here you see that actually sentences from the planner in some way are executed by our robot. And here you see, of course, and there the temporal information is important. Good, some reflections. So what do we know about the primate's vision, which is relevant for engineers? So the first thing is the richness of the representation. So we have a zoo of features here, a huge variety of different aspects is processed on different levels of abstraction. And the challenge is, how can we design or learn such a rich representation? One word about deep hierarchies versus flat hierarchies. So this is clearly a deep hierarchy. So a flat structure would be more something like this, where which has been quite common in computer vision. You do some basic feature processing, and then you have a task-dependent neural net that is doing something. But here in a deep hierarchy, you share features, so different tasks share features on lower levels. And I also want to point out that a deep hierarchy is not, so there are also biological systems that don't establish a deep hierarchy. The most famous example is probably the frog, where there are fly detectors in the retina. So this is an example. So biologically, doesn't mean necessarily deep hierarchies. It depends very much on the being. What we have is an increasing receptive field size in general, and an increasing level of abstraction in the processing. Flat hierarchies are inefficient, I would say, because they don't share computational resources. And also I believe that when different tasks are solved within the same representation, then the transfer between tasks is easier. One of the core issues for cognitive agents, and that was it basically. Thanks.
Thank you, Norbert. Any questions from the floor? Question at the back there.
Hi, yes, thanks for the very interesting summary. I didn't hear any mention of feedbacks in pathways. For example, there's even feedbacks all the way to the LGN, I believe.
Yeah.
Is that something you're also trying to emphasize?
Yeah, it is briefly discussed in the paper, and of course. But I must say, this is one of the, I mean, we know that there are even more feedback connections than feed-forward connections, but we know very little about what they do, except in a very abstract sense, that they're probably used to disambiguate information and so on. And therefore we didn't dwell so much, because we know it is there, we know it is very important, but we don't know much about the details there. Yeah, but I fully acknowledge that, yeah. But it's better to write about things where we know something than about things we don't know so much about.
If I use knowledge of artificial neural networks where there is a loss function and you want to minimize some kind of response to the network, what do you think the visual context is doing when it's learning? What kind of cost functions to minimize?
I mean, there has been the attempt, and I think they went quite far, to learn actually these features actually on the level of V1, let's say, by optimizing sparseness or reconstructability or slowness or something like this. So that would be one indication. I believe that another important thing, since I see the visual input, it triggers something where then all the stored knowledge that we have about scenes comes in to come to a very reliable scene representation, although the underlying signal is extremely weak. So I think that's one of the arts that the vision system can do. And now I have forgotten what I want to say. You asked about the objective function. Yes, and I believe another important objective is predictability. So we want to have features that are able to predict events at other parts of the visual cortex, because that is required for disambiguating the visual information.
One last question, right there.
Yes, very interesting talk. I was wondering about the interaction between the visual cortex and the so-called mirror neurons. Is there something that you could add?
Yeah, I've not a good answer to that. These mirror neurons have been found in areas that was above the areas where I was talking about, in this kind of more mixed areas. But my feeling is it's probably an independent concept of vision. Actually, to be honest, I don't have a good answer to your question.
You might have an opportunity to ask that of Marco when he does his keynote talk later on in the day.
Good. Okay, thanks.